So we covered the first part of the central dogma of biology. We covered how DNA gets used as a template to make RNA. We covered how in eukaryotes, not in prokaryotes, how messenger RNA is processed in the nucleus with a few different modifications. We talked about the 7-methylguanosine cap, we talked about the poly A tail, we talked about RNA splicing, but how does all of this go into making a protein? Well, all those modifications allow the mRNA to get exported from the nucleus and to survive out in the big scary cytoplasm for a while, for long enough to be translated before it is destroyed. And translation is the process of making RNA into protein. And this is going to involve all three kinds of RNA. So we've established that messenger RNA is going to be part of this. Messenger RNA, or mRNA, is going to be the template that we read in order to make protein out of it. So it's not that RNA gets turned into protein, but more that RNA serves as a recipe from which protein is made. Then we're going to have ribosomal RNA. The ribosome is a complex of our RNA, or ribosomal RNA, and proteins that is going to help us make the protein. It kind of looks like a hamburger. It's got two parts. It's got its large subunit on top and the small subunit on the bottom. So that's these guys, large and small subunit. And one thing that you might have to know if you're in college level biology is that the ribosomes in eukaryotes and prokaryotes are a bit different, and this is based on the densities of the RNA. And these densities, we're going to measure them in something called Svedberg units. So if you're in a lab, if you've ever used a centrifuge, centrifuges are going to bring things down based on their mass and based on their density. And you can have a tube with a density gradient. So something that's really not dense on the top, and as you go down, the liquid gets more concentrated and it gets more dense. So if you centrifuge something, say ribosomes, for a while, the more dense things are going to fall down and they're going to go to the lower layers. And in this way, people have made this system where they use Svedberg units to describe density as shown by centrifugation. So in eukaryotes, this is going to be an 80S ribosome. That's just the density number is 80. And the large subunit is going to be 60 and the small is 40. Nobody ever explains to you in classes exactly why these numbers do not add up. But if you think about a density as a ratio between mass and volume, so it's not likely that it's going to be additive the way that mass is. In prokaryotes, you're not going to have 80S ribosomes, you're going to have 70S ribosomes. So these are going to be made out of 30 and 50S subunits. And these kind of things are actually really cool because there are antibiotics that target bacterial ribosomes because these bacterial ribosomes are different than eukaryotic ribosomes. Or in evolutionary biology, the reason that we know that archaea and eukaryotes are more related to each other than archaea are to bacteria is that bacteria have these 70S ribosomes, but archaea actually have 80S in the way that eukaryotes do. Another application of this is the endosymbiont hypothesis. So the endosymbiont hypothesis says that mitochondria and chloroplasts were once some form of prokaryote or some form of bacterium, I guess, that entered the eukaryotic cell through endocytosis and evolved in a symbiotic relationship to become the organelles they are today. So the evidence for that, besides the circular chromosomes in these organelles and a bunch of other stuff, is that they have 70S ribosomes. The mitochondria and the chloroplasts and this other organelle called the hydrogenosome all have ribosomes that resemble bacterial ribosomes, and that's one of the pieces of evidence that formed the endosymbiont hypothesis. Anyway, ranted out ribosome densities over. The third RNA that's involved in this besides mRNA and rRNA is called a transfer RNA or a tRNA. And the tRNA's job is to connect the amino acid to the RNA code. So every 3 bases in our RNA is going to represent one amino acid. And every 3 bases, we call that a codon. So let me see what codon this one is. It is AAG in our little diagram. On our tRNA, the tRNA is going to have an anticodon, so that's going to base pair with the codon. So in this case, if we had a messenger RNA that was right here, it would be UUC, right? Because RNAs have U instead of T. That pairs with A. So this is our codon. And then the tRNA is going to bind to this, facilitated by the ribosome with its anticodon, which is going to be AAG, as shown here, right? So each of these bases is going to pair with each other and recognize each other. And the reason this is important is because at the other end of the tRNA is an amino acid binding site. And this is the amino acid binding site. And every tRNA that has a specific codon, or I mean a specific anticodon, is going to be associated with a specific amino acid. There's actually going to be more than one anticodon and more than one codon per amino acid. So this code is slightly redundant, and that's kind of important for evolution and for mutations, but I'm going to talk about that later. These tRNAs are going to gain their amino acid through a little enzymatic sequence. They're going to work with an enzyme called amino acyl tRNA synthetase, and they're going to use up ATP, so this process requires a bit of energy, but the amino acid is going to stick onto this transfer RNA, and eventually the amino acyl tRNA, or the tRNA that is attached to its respective amino acid, is going to find its way into the ribosome. And it's going to join with the messenger RNA, and the whole thing is going to work out. So all these codons and anticodons that match with amino acids can be summarized in something we call the genetic code. And this is often represented in this three-way table that's shown here. There's 20 amino acids, right? And 4 base pairs, and every 3 base pairs are going to represent a code for an amino acid. And we might think, okay, well, combinations of 3 base pairs, that's way too many to be 20. Well, that's actually right. There's way too many to be 20. Like I said, the amino acid, the genetic code is redundant. So you're going to have more than one, codon or base sequence from nucleotides to represent one amino acid. And this is kind of convenient because let's say that we have a mutation, which is a change in your DNA, which is going to reflect in a change in your RNA. So it's a nucleotide replacement or something like that. Let's say that we had this CCU as a sequence in a nucleotide. So this would be our RNA that came from our DNA. So our DNA might have been GGT was our sense strand or the coding strand of the DNA. And this is going to make our CCU in our RNA, right? Let's say that this position got mutated. Let's say this position got mutated to CCA. Then this would still work, right? Because CCA is still representative of pro, which is proline. This might be your time to get familiar with the 3-letter amino acid codes as well as the 1-letter amino acid codes. I have a video on amino acids. I would have to go back and see if I went over the 1-letter and 3-letter amino acid codes, but that is basically memorization, and there's a couple tricks to memorizing them. But yeah, get familiar with those, and this table will make a lot more sense. There are a couple unique kind of quirky codons in here. There's a start codon, that's here in green, and all RNAs that get made into protein, there are some non-coding RNAs. We can talk about that in another video. But all RNAs that get made into proteins start with AUG. And AUG codes for methionine, which is an amino acid that looks like this. So that's methionine, and that's what all of these are going to start with. There's also a few stop codons. So these guys are going to tell this whole ribosome and tRNA and mRNA and everything that goes into making protein, you guys are free to go. And when we talk about mutations, we're going to see that if we get a stop codon and it comes way too early in the sequence, that's a huge problem, right? Because it's going to cut off a whole piece of our mRNA and consequently a whole piece of our protein. So how do we start this whole process? Things are going to come together one by one. So the first thing that's going to happen is that our mRNA, our messenger RNA, is going to bind to our small ribosomal subunit. So this is going to happen, and then we are going to get what's called an initiator tRNA. So like I said, every sequence of protein is going to start with methionine. If you go in and you look at like different protein sequences, this might not always be true of the end product because proteins do get modified afterwards. So like if you are in research and you see a protein and it doesn't have methionine on the start, don't freak out. Proteins get cleaved, proteins get modified, but when they are initially getting synthesized, all proteins are going to start with this initiator tRNA that codes for methionine. In prokaryotes, so in bacteria and in archaea, they've got this special modification where they are going to have formylmethionine. And remember that formaldehyde is a 1-carbon aldehyde. So formyl is going to be 1 carbon with 1 carbonyl group added onto this methionine, and that's going to help the bacteria and archaea recognize that they have to do this. This starting step is going to expend 1 ATP, so this needs energy, and the large subunit is going to bind, and we're going to start. And things are about to get really complicated because we have these 3 different sites where these tRNAs can bind. But we're going to make sense of it, okay? So the first thing that happens is we have a tRNA with a growing polypeptide, and that's what we just call a polymer of lots of amino acids that maybe is not yet a fully formed protein. So that's going to be in this middle site. And we've got names for each of these sites. This site where the polypeptide is, we call that the peptidyl site. So this is going to be P. I hope that makes a bit of sense, right? Oops, I drew a random line. Okay, so the next thing that's going to happen is the mRNA is going to kind of move along, and we are going to read our next codon with our next tRNA. So this new tRNA just came in, and it's coming into a site known as the A site, and the A is for aminoacyl, right? So our peptidyl site had a polypeptide on it, or a growing polypeptide, and the aminoacyl tRNA site has a tRNA with 1 amino acid. So the names make sense so far. The next thing that's going to happen is this polypeptide is going to switch places. So this whole thing that was growing already is going to bind to the amino acid. So this is going to go here. So it's going to switch which tRNA is attached to. So now this one doesn't really have anything to do, right? It already had its time in the spotlight. It was a polypeptide. It gave its polypeptide, or its growing polypeptide, to the new RNA that came in, the new tRNA. And now this one, it's time for it to go and make room for a new one. So it is going to move. Both these tRNAs are going to move because now this one is not aminoacyl anymore, right? It's peptidyl. It's got a polypeptide on it. So it's going to move to the P site from the A site. And the old one that doesn't have anything on it anymore, it's going to move from the P site to something we call the E site. And E is for exit. So this one is going to end up exiting the ribosome. And now we're back to where we started. We have a polypeptide growing on the peptidyl site, and we have the aminoacyl site open for a new aminoacyl tRNA to come in. And this is going to happen, and it's going to keep going, it's going to keep going. And just as we saw in transcription, remember in transcription we had initiation, we had elongation, we had termination. Here we had initiation, that was up here. We have elongation, that's this whole process where the polypeptide is growing. And then termination will be the ribosome will come across a sequence of RNA that tells it to stop. And the sequence of RNA that tells it to stop is going to be one of these stop codons here. The ones in red that I circled, these guys. And when it gets to one of these guys, the tRNA that comes in is going to have a release factor. And the release factor is going to cause this whole assembly to dissociate and go bye-bye. So these ribosomes can occur in basically 2 conditions. One of them is called free ribosomes, and free ribosomes just kind of float around in the cytoplasm. They're not attached to anything, and they make proteins that are not associated with any kind of membrane. Another kind of ribosome is called a bound ribosome, and you might remember if you talked about parts of the cell. I haven't made a parts of the cell video yet. That's something I have to do, but remember that the endoplasmic reticulum has 2 parts, rough and smooth. So I think this is the nucleus. this is going to be the endoplasmic reticulum, and I think the book I use draws it in blue, so I'm going to draw it in blue. The rough endoplasmic reticulum is called rough because it is covered in ribosomes. So these ribosomes are going to synthesize proteins on the outside of the endoplasmic reticulum, and there's little pores in this organelle, and the proteins that it makes, they're either going to end up embedded in the membrane, and then they're going to bud off in vesicles, which are like little packages of membrane that can travel throughout the cell and even be secreted outside of it. Or they can be secreted into what is known as the lumen, or the inside of the endoplasmic reticulum. So then they're going to be inside a compartment. So free ribosomes out here, they're going to make proteins that are on the outside of an organelle. They're going to be inside the cytoplasm, but they're going to be outside an organelle. Bound ribosomes are either going to make things that are transmembrane, so they're going to be going through a membrane with part of it like embedded inside the phospholipid bilayer, or they're going to make things that go in the lumen of the ER. And these things, like I said, they can bud out in a vesicle. So if you look at this diagram, here's our vesicle, it's coming off, and eventually it's going to become its own little entity, right? And it's going to have this protein inside of it. And a couple things can happen with this. If we've got another organelle, like say that we were making a protein, and this protein is a digestive enzyme, right? We can fuse this vesicle with the lysosome, and we can release this protein into the lysosome to perform its hydrolytic function, right? Or we can have, if this is the outside of the cell, this is the plasma membrane that separates the cell from everything else around the cell, this can get secreted. So I have yet to also make a video on endocytosis and exocytosis, but exocytosis is the process by which, if you have a vesicle inside of the cell, it can fuse with the membrane and release its contents on the outside of the cell. So say this protein is a hormone, or maybe not a protein, maybe it's just a polypeptide. There's things called peptide hormones, and that's like adrenaline. Adrenaline is a peptide hormone. If you have adrenaline in this little vesicle, and it's a bound ribosome that secretes it into the lumen of the endoplasmic reticulum, and then releases this as a vesicle, and then outside of the cell, you can secrete it outside the cell membrane to go into the bloodstream to all your organs that are affected by adrenaline. So those are a couple of fates that a protein can take. One more thing that's important in the function of ribosomes and the synthesis of proteins through translation is that it doesn't happen in a vacuum. You don't have just one ribosome living in one cell. You've got lots of them, right? So in prokaryotes and in eukaryotes it's going to be more the free ribosomes. In prokaryotes, you don't have an endoplasmic reticulum, so they're all free. You can have an mRNA transcript. I think they draw it in red in the book that I'm working with, so I'm going to draw it in red. And they've got multiple ribosomes. You can have multiple ribosomes translating the same mRNA at the same time. And they can all be making proteins at different parts of the mRNA. So if there's a book series, say that this is me, I'm reading the Harry Potter series, right? If I'm reading the Half-Blood Prince, when I'm done with the Half-Blood Prince, I can pass it on to my younger sibling, and she can read the Half-Blood Prince while I'm reading the Deathly Hallows up here, right? So I hope that makes a little bit of sense that multiple ribosomes can translate the same mRNA and make the same protein out of it at the same time, just in order, so they're at different places in reading it. And that's translation. So we've finished our little saga of the central dogma of biology.